Hello, welcome to Physionic, where we learn the body from the macro to the micro. In this piece of content, we will be investigating some science on how our cell's skeleton interacts with mitochondria to allow them to split into two, otherwise known as mitochondrial fission.
Learn your body, a science-based education.
As forewarning, this content is heavily cell and molecular biology focused, but I hope that by me supplying the study and my notes breaking down the terms and offering more details, you will follow along and learn something with me. Now, a bit of background on cell biology and the major players in this study. Mitochondria are organelles that are found in many of our cells, and they have many functions within our cells. That said, our cells also have a cytoskeleton, or cell skeleton, made up of proteins called actin. These single proteins, called globular or G-actin, will string together into filaments known as filamentous actin or F-actin. This actin protein assembles and disassembles between G and F-actin continuously to allow the cell to move as one of its primary functions. However, there are many undiscovered functions of actin, and one of those functions, as we might see, is its association with mitochondria as they undergo mitochondrial fission. Mitochondrial fission is a cool phenomenon wherein a mitochondrion will pinch in the middle until it splits into two baby mitochondria. Actin may facilitate that fission in some capacity, so let's see how. Okay, for mitochondrial fission to occur, key proteins have to attach with the mitochondrion that is aiming to split into two. One of those key proteins is DRP1, which is critical for mitochondrial fission to occur. So the researchers have taken images of cells and are fluorescing red for the F-actin. Remember, that's filaments of single actin proteins, and are fluorescing green for mitochondria, and they want to know if they interact. You can tell if they interact if the two colors, red and green, are in the same location, because the color will change to an orange-like color. Interestingly, the researchers looked at control cells without any manipulation. Then again at those cells, but they add a fission-stimulating drug, and then they repeat both experiments with and without the same drug, but in cells that do not express DRP, the key fission protein I mentioned. You can see two things here. One, that cells that are not stimulated to undergo mitochondrial fission by a fission drug do not have the interaction of F-actin to mitochondria. Second, knocking out, eliminating DRP from the cells still allows actin to associate with mitochondria, but they do not undergo fission. Hence why they look elongated compared to cells under the same conditions, except for their expression of DRP. Now we see that DRP is essential for mitochondrial fission, and we see an association between actin and mitochondria, but we still don't know if inhibiting actin would stop or inhibit mitochondrial fission. So, with this final piece of data, the researchers are inhibiting the cells from producing a necessary actin-regulating protein called cortactin. Cortactin is one of the proteins that facilitates the polymerization or the conversion of G-actin to filaments, the aforementioned F-actin. So if it is inhibited, actin cannot switch from G to its filamentous F form. So what will this do to mitochondria? Here, the researchers have inhibited cortactin and added the fission-inducing drug, and are comparing it to a control wherein they did not inhibit cortactin, an otherwise healthily functioning cell. As you can see, the mitochondria of the control cell are fragmented as they split into smaller mitochondria, aka fission, due to the stimulus of the drug. However, when actin is unable to form F-actin, the mitochondria remain elongated and unable to form their split, smaller versions. Here is some evidence that actin is likely a key player in mitochondrial fission. So, in conclusion, the main conclusion of the study shows that actin is a crucial factor in allowing mitochondrial fission, or the separation of one mitochondria into two to occur. We've looked at two pieces of data to come to that conclusion, but rest assured, there is much more to this study. Still, these two pieces of information are poignant and drive home the conclusion. I hope this proved interesting and informative, and with that said, keep your actin functioning and your mitochondria will healthily fission for you. Until next time, cheers.